genetics and evolution. Let us move on to the next chapter, chapter 6. Molecular basis of inheritance. So far, we have been discussing about different patterns of inheritance, different kinds of variations and all that. At the molecular level, how this inheritance takes place. After Mendelism became popular, there was a search for what is the substance in the cell that is responsible for this pattern of inheritance. So, there was a search for the genetic material. Mendel used the word factors. Factors being responsible for the expression of different characters. Subsequently, these factors were identified as genes. But when Mendel conducted these experiments and said that certain factors are responsible, there was no idea about what was that factor, in what form does it exist in the cell, and so on, because cell division was not known, formation of gametes was not known, even chromosomes had not been discovered. Therefore, nothing was known at the time of Mendel. But subsequently, chromosomes were discovered, and as we have discussed earlier, the chromosomal theory of inheritance was established. So, scientists started looking for some substance which may be present in the chromosomes that can function as the genetic material. In 1869, a German biologist by name Miescher extracted a chemical substance from the nucleus of a cell and named it as nuclein. A little later, this nuclein was found to contain an acidic substance, but its exact chemical nature could not be established and its role in the cell could not be established. So, scientists went on searching for the genetic material, and they were very, very clear in their idea that any substance which functions as the genetic material should have 4 specific properties. 4 specific properties. 1, it should have the capacity to make its own identical copy. If the same character is to be expressed in the next generation offspring, that genetic material should be able to make its own identical copy. This property is what we call today as replication. Number 2, this chemical substance should be comparatively stable. It should not undergo very frequent changes. Changes, if any, should be very, very rare. Number 3, that chemical substance should contain the genetic information in some form. It should contain the genetic information necessary for carrying out the cellular functions. For carrying out the cellular functions. And number 4, it should be able to transmit this genetic information to other parts of the cell. So any substance which can satisfy these 4 conditions can function as a genetic material. This was the idea working in the minds of scientists. Finally, in the year 1928, a biologist by name Griffith conducted an experiment working with bacteria Streptococcus pneumoniae. Streptococcus pneumoniae, the bacteria which causes pneumonia. His experiments gave a clear idea about what could be the genetic material in the cell. This bacteria, Streptococcus pneumoniae, occurs in several strains. These strains are called 1, 2, 3 and so on. Strain 1, strain 2, strain 3 and so on. When these bacteria are cultured, the colonies, resulting colonies, contain bacteria that can be of two types, some capsulated and some uncapsulated. The capsulated bacterial cells developed a shiny, smooth capsule made up of a particular polysaccharide, whereas the uncapsulated bacteria had a rough covering. So these bacteria with a shiny, smooth capsule were called S strain, and these bacteria with a rough covering were called R strain. He used both these bacteria and injected these bacteria on the mice, experimental mice, and found out specific results. Let us see what was that experiment.